Hi, my name is Luke Gilbert. I'm an assistant professor at the University of California, San Francisco. I'd like to thank the organizers for this opportunity to talk to you today. My title is Revealing Cancer Dependencies and Determinants of Therapeutic Response Using CRISPR. I think we can all agree that biology has undergone a genomics revolution over the last 25 years or so, where we started with sequencing a single human genome to define the set of genes that are encoded by the human genome, and then over the last several decades have now sequenced many thousand human genomes in healthy individuals and in a variety of disease states. In cancer biology, this has resulted in an advanced draft genome of the cancer genome, where we now know the set of mutations, amplifications, and deletions that are associated with many common types of human tumors. And in some cases, that's led to clinical hypotheses and breakthrough therapies for treating cancer patients. But in other cases, we don't know enough about the genes involved or implicated in a disease subtype to be able to translate this directly into new medicines. And so I think in parallel, there's been a revolution in what I would call functional genomics, or our ability to experimentally characterize genes. And this has been driven largely by CRISPR over the last 8 or so years. And what we hope is that functional genomics, together with clinical genomics, can objectively and systematically map how mutations drive cancer progression and lead to new insight, which promotes new medicines for the treatment of cancer patients. And so today, I'll tell you about my lab's efforts in CRISPR functional genomics applied in 3 areas. The first is to identify genes required for specific cell states. The second is to reveal determinants of response to new drugs and nominate innovative new combination therapies. And the third is strategies for targeting loss of function driver mutations. And so in the first set of slides, I'll tell you about our efforts to characterize advanced prostate cancer driver genes, such as the androgen receptor. So for those not familiar with prostate cancer, this is a common disease in men. It's driven largely by a dominant oncogene called the androgen receptor, which is a transcription factor. The androgen receptor is cytosolic until it binds to its hormone ligand, testosterone, at which point it translocates the nucleus to drive gene expression of genes that promote prostate biology and also prostate cancer and prostate cancer progression. And so we are interested in learning new things about how genes regulate androgen receptor biology and disease progression in advanced metastatic castration-resistant prostate cancer, which is the disease that most people die of in this cancer type. And so to do this, the first thing we did was we created several prostate cancer CRISPR-I models that express stably our CRISPR-I construct that silences gene expression. We can then generate lentiviral genome-scale CRISPR-I sgRNA library and infect this into cells. We then conducted a cell proliferation screen where we allowed cells to grow for about 10 population doublings, and then we can count sgRNA abundance in our T0 sample and our endpoint to identify genes that regulate prostate cancer cell proliferation. That gives you a volcano plot that looks like this, where we identify between 1,000 and 3,000 essential genes that are required for prostate cancer progression. And these are highly reproducible phenotypes, but they're not that informative in that the vast majority of these genes are required for all human cell proliferation. And so our goal was to design an informatic pipeline which would discriminate between common genes that are required for cell proliferation and prostate cancer-specific driver genes. And so to do this, we took our CRISPR-I screen data from 2 cancer models, and we filtered using the list of common essential genes provided by DepMap and Project SCORE to remove common essential genes required for all cell proliferation. We can then cross-filter against in-house CRISPR-I data in other solid tumor models and leukemia models to remove false negatives associated with CRISPR versus CRISPR-I. And that left us satisfyingly with a list of about 34 prostate cancer driver genes. And reassuringly, the number 1 gene on this list was the androgen receptor. And so we think this is a straightforward pipeline for identifying cancer-specific genetic dependencies in really any cancer, but in this case in prostate cancer. So we can take these genes nominated by our screens and perform follow-up experiments. In this case, this is a GFP competition assay where we can look at how knockdown of individual genes affects cell proliferation over a time course. And what you can see relative to a negative control is that, as expected, the androgen receptor is indeed required for cancer cell or prostate cancer cell proliferation. We see that other top hits, including HOXP13 and grainy head-like 2, are also similarly required for cancer cell proliferation. And then we see genes that have not been previously implicated in prostate cancer biology, such as KIP4A, that are sort of equally required for prostate cancer proliferation. And so this is exactly what we wanted to see, in that we can nominate genes that have robust phenotypes for both positive controls and unexpected genes. So how specific are these novel genes such as KIP4A for prostate cancer proliferation or survival? To answer this question, we did the following experiment where we used 10 cell lines, 5 prostate cancer cell lines, and 5 non-prostate cancer cell lines, and we knocked down KIP4A and then did a calling formation assay to look at whether KIP4A was required for cell proliferation or survival. And we can see that in all 5 of our prostate cancer models that are both AR dependent in green and AR independent in gray, KIP4A is required for prostate cancer proliferation and survival. In contrast, 4 out of the 5 non-prostate cancer models, which are representative of a diverse set of tumors, showed no dependency on KIP4A at all. And so we think that this is reasonable validation that KIP4A is a relatively selective prostate cancer driver gene. We looked more into the clinical data on KIP4A to try to decipher some of this biology from human samples. And so what we see is there's no relationship between KIP4A levels and the mRNA levels of the androgen receptor. And so this suggests in an indirect way that KIP4A is not, together with our data on the previous slide, is not driving prostate cancer progression in an AR-dependent manner, but instead is an AR-independent prostate cancer driver gene, which I think is quite interesting in and of itself. And then we can show that, as would be expected for a cancer driver gene, that KIP4A expression stratifies patient survival. And so if you look at high KIP4A expressing tumors, these patients do quite poorly relative to KIP4A low patients. We can validate KIP4A as a prostate cancer driver gene in vivo as well. So this is data where we used an inducible CRISPR-I model, and we can implant cells expressing either negative control guide RNAs or KIP4A guide RIs. We knock down KIP4A specifically in vivo, and we see that here in this assay, KIP4A is also required for disease progression in vivo. And then lastly, if we overexpress KIP4A in benign prostate models, shown here in these blue bars, so RP1, PNT2, we show that KIP4A overexpression drives colony formation in these non-malignant prostate cancer models. And so collectively, these data suggest that KIP4A is indeed, as nominated by the screen, a prostate cancer driver gene that's relatively selective for prostate cancer. And so this is exactly what we wanted to see. We're seeing functional genomics identify known and new targets and learning new things about driver genes that may be dependent or independent on the androgen receptor. To study androgen receptor biology more directly, though, we decided to take a second strategy in which we fluorescently label the endogenous AR protein. And to do this, we leveraged a technology that was previously developed at UCSF, where we can tag genes using split fluorescent proteins. And so we optimized our protocol using a split GFP tagging Sec61 beta, and then we used this protocol to tag the androgen receptor endogenously using a split neon green construct. We spent about a year characterizing these models to show that the tagged androgen receptor does not perturb androgen biology in any way that we can measure. And then with those data in hand, we set out to do a genome scale flow cytometry screen to identify genes that directly regulate AR protein abundance. So to do this, we used our same prostate cancer CRISPR-I models. We can put in the genome scale sgRNA library, expand cells for a few days, and then harvest cells and use a flow cytometer to sort cells that have low AR protein expression or high AR protein expression. And then we take these cells, extract genomic DNA, and count sgRNA abundance within each fraction by deep sequencing to identify genes that dictate androgen receptor protein levels. And this enables us to systematically identify genes that are negative regulators of androgen protein levels, as well as genes that are positive regulators of androgen protein levels. And so again, here's a volcano plot displaying the results of this genome scale CRISPR-I screen. And what we can see to our satisfaction is that, as expected, the androgen receptor itself is the very top hit in this screen. So if you knock down AR with CRISPR-I, which decreases AR transcript levels or transcription, this results in decreased AR protein as expected. And so that was a nice positive control for these kind of facts-based screens. And then when we look at other top hits, we can see again that we see both known and new regulators of AR protein activity or abundance. And so these are genes like HOXP13 and grading head like 2 and GATA2, which are known to be AR transcription factor co-regulators and support AR activity in prostate cancer models. And so this, together with additional hits that are quite novel, suggests that we can use both simple cell survival and proliferation screens with additional filtering steps, or flow cytometry-based screens to identify key regulators of the androgen receptor and the prostate cancer cell state. And so we think we're quite excited about this coalescence of multiple screen formats to narrow in on gene networks that drive specific cell states. The second thing we were interested in doing using CRISPR functional genomics was revealing determinants of response to new drugs even before they hit the clinic. And so we saw with great interest that our colleague at UCSF, Kayvon Shokat, published a new class of KRAS G12C inhibitor small molecules a couple of years ago. And so we wondered whether we could use genome scale screens to identify genes that regulate response to this small molecule, even just as clinical trials are being initiated. As background, just to keep everybody on the same page, RAS is a GTPase, so it exists in two forms. In the RAS GDP bound state, it's off. The RAS is off and does not promote pro-oncogenic signaling. In the RAS GTP bound state, RAS is on, and in this state, it binds to downstream effector proteins such as RAF and PI3 kinase. And what I think many of you will know is that KRAS is one of the most commonly mutated driver oncogenes in cancer. It's frequently mutated in lung cancer, pancreatic cancer, colon cancer, and more. And what happens with these mutations is that they lock RAS in the on state, so in the GTP bound state. And this results in constitutive activation of downstream signaling pathways through RAF and PI3 kinase and others. And so that all sets the stage for this new small molecule that Kayvon invented. What's key to know is that this small molecule only binds to KRAS in the GDP off state. And so that represents a vulnerability or a liability for these small molecules, in that we know that the majority of RAS protein in patients that have activating RAS mutations is in the GTP on state, but the drug only binds in the GDP off state. And so that this was the primary motivation for the screen. We wanted to identify genes that could drive an increased pool of the RAS GDP bound state, which would then render cells and tumors more susceptible to these RAS G12C inhibitors, promoting therapeutic outcomes in patients. And so, same as for the prostate cancer example, we start always by creating cell models of the disease state we want to look at. So we created two CRISPR-I models of RAS G12C driven cancers. In this case, one is a non-small cell lung cancer cell line and the other is a pancreatic cancer cell line. We validated these and expanded them, and then we can put in our genome scale CRISPR-I sgRNA library, expand cells for a few days, take a T0 sample, and split the remaining cells into an untreated and drug-treated arm of a screen. And we treat with this RAS G12C inhibitor for about 10 days. And then we harvest cell populations and count sgRNA abundance in our T0 untreated and drug-treated samples to quantitatively and systematically identify genes that regulate response to a KRAS G12C inhibitor. So if you do this from both cell models and sequence the results, what we saw shown in these waterfall plots are that we identify KRAS itself and a number of other critical genes in the RAS pathway, indicating that the screens are working as expected to identify genes that regulate response to a KRAS G12C inhibitor. We also see in teal that there were a variety of other shared hits between these two screens, and these cell lines are quite different, right? Just to remind you, one's a pancreatic cell line and one's a lung cancer cell line. So it's really interesting to us that we do identify such a large number of shared hits between both cell lines in areas of biology that aren't normally associated with RAS biology and RAS signaling. What you might be wondering is how this work is different from work over the last decade or so on KRAS synthetic lethal phenotypes. In this case, we're not, those phenotypes, KRAS synthetic lethal phenotypes, are looking at genes that are required for cancer biology in the context of KRAS being activated or on. In this case, we're looking at genes that are required for cell survival or proliferation in the context where we've acutely turned off KRAS signaling. And so this is not a canonical synthetic lethal relationship, and instead we've called these collateral dependency genes. If you look at this data in another format, and this is the pancreatic cancer data, we see a lot of expected genes that we would have predicted modulate response to this drug. And so these are genes that are upstream of KRAS signaling and modulate the like receptor tyrosine kinase biology, and also the guanine exchange factors that promote KRAS GTP levels. And so if you knock the genes like that down, you sensitize cells to the KRAS G12C inhibitor by increasing the cellular pool of KRAS GDP. Moving towards the idea of constructing combination therapies that would increase response to a KRAS G12C inhibitor, we looked at these lists and identified a number of genes that we predicted are druggable and or have FDA-approved drugs, where combining a KRAS G12C inhibitor together with drugs targeting sensitizing phenotype genes from our screen, we predicted should result in drug synergy and increased patient response. And so we presented a number of these in the paper, and this work's actually published. So I'll just present one that didn't make it into the paper, but we're really excited about. And so this is a patient-derived xenograft, or PDX, which is it's a non-small cell lung cancer PDX that's RAS G12C mutant. It's a homozygous mutation, but despite this, the PDX is completely resistant to this KRAS G12C inhibitor. This is something that's being seen currently in clinical trials. And so that's the data shown here in black versus gray curves, where when we initiate G12C drug dosing, we don't see the tumor respond at all. Similarly, this tumor is almost completely resistant to a CDK4/6 inhibitor. CDK4 was one of our top sensitizing hits from both genome scale CRISPR-I screens. And so what we predicted from our CRISPR-I screens is that if we combined both a CDK4 inhibitor and a KRAS G12C inhibitor, we'd see a synergistic response and tumor regression. And that's satisfyingly exactly what we see here, where we see almost complete tumor regression in the combination therapy with a pretty durable long response even after we end dosing. And importantly, we don't see this combination therapy being toxic. And this is in large part because the KRAS G12C inhibitor only binds to a mutant protein, and so it has very low toxicity on its own, enabling it to be leveraged into combination therapies unlike previous approaches. So lastly, I'd like to tell you about our strategies for targeting loss-of-function driver mutations. And this is really important because you can't directly target a protein that's missing. We need functional genomics to identify how loss of a gene rewires cell signaling to promote increased dependency on other pathways. So for this portion of the talk, we'll be talking about acute myeloid leukemia. Acute myeloid leukemia is an aggressive leukemia. In its high-risk forms, adult older patients only have a median survival of about 4 or 5 months. But what people have realized over the last couple of years is that these patients do respond to some degree to a drug called decitabine, which is at low dose and thought to be a hypomethylating agent that inhibits DNMT1. But this drug is really very poorly characterized, and so I just highlight this New England Journal of Medicine paper that says that the background is the molecular determinants of clinical response to decitabine is unclear. And so we wanted to get a sense of how this drug is working. And one of the most interesting things from this paper was that patients with a p53 mutant tumor genotype respond as robustly as patients without p53 mutations. And this is a really unusual relationship. Across almost all cancers, loss of p53 promotes tumor progression and drug resistance. And so this suggests to us that decitabine is somehow killing AML cells in a p53 independent manner, and we were curious to study this and identify new strategies for drugging loss of function mutations, as for example in p53. So to do this, we again created an AML model of high-risk leukemia. We put in our CRISPR-I machinery, and then we can expand the cells and transduce with a genome scale library. And then we split the genome scale screen into 2 arms, where we either treat with DMSO or this drug we're interested in, decitabine, for about 10 days. Then we extract sgRNAs and deep sequence to identify genes that modulate response to decitabine. That again gives you a volcano plot like this, where we've systematically elucidated all genes in this model that control response to decitabine. And what we see is, as expected from clinical data, we see genes like BCL2, which are key regulators of cell death show up as strong sensitizing hits, suggesting our screen is on the right path for identifying genes that are important for response to cytidine and p53 mutant samples. And then really interestingly, and unexpectedly, we see a large number of our top sensitizing hits are genes that regulate RNA quality control pathways. And so we can validate these RNA quality control pathway synthetic lethal phenotypes with decitabine, for example, in this graph where we can combine an RNA decapping inhibitor and along with decitabine and show that we get synergistic induction of caspase dependent cell death. So the data here is for caspase 3-7 activation in cells over a dose response curve. And we think that this is a really viable clinical strategy for treating AML patients where you can combine these RNA quality control inhibitor drugs that are surprisingly very non-toxic together with decitabine to increase patient survival. And mechanistically, what we think is going on here is that DNA hypomethylators put a huge amount of stress on the cell through dysregulated transcription and potentially splicing, which then results in an induced synthetic lethality with RNA quality control checkpoints and RNA decapping activity, such that if we induce both at the same time, the cells are unable to cope with this. So with that, I'll end and just say thank you for your time and attention, and for a bunch of people in lab who did all the work and funding, and thanks, and I'll take questions.